Hello and welcome back. We're going to continue on the topic of gene transfer in prokaryotic cells. In a previous video, we discussed the processes of conjugation and transduction in transferring genes to bacteria. The third method of gene transfer we're going to cover now is transformation. And transformation is really kind of neat because it is used a lot as part of biotechnology. So it's used at research universities, it's used in pharmaceutical companies. And what the biotechnology industry is doing with transformation is they're taking advantage of the fact that a single gene can be taken from one organism and transferred to bacteria. In this case, the example I'm going to use for our single gene is going to be insulin. Now, there are a lot of other genes that are made, or proteins that are made this way, because what you want to keep in mind with bacteria, just like eukaryotic cells, they have the same flow of genetic information. They take DNA, they transfer that information from the DNA into RNA. That RNA is then used to build a protein. So what we're doing in biotechnology is we're taking advantage of some basic cellular processes that go on in prokaryotic cells to make something we want. Now, with this process, okay, you'll also see it referred to as recombinant DNA technology. When something is recombinant as far as DNA, what that means is you have two different genomes, or you're involving two different genomes. In the example I'm using here, we're going to have human DNA in the case of the insulin gene, and we're going to have the bacterial genome, okay? So when you make a protein in another organism, that protein is referred to at that point as recombinant. Oh, I can spell recombinant. I did it before. All right, so recombinant. What happens when that DNA is transformed to the bacterial cell? We're going to take our donor DNA. In this case, it's going to be a plasmid, a bit of circular DNA that has the insulin gene on it. And we're going to take bacterial cells that are deemed competent. So in the biotechnology world, what they do to make cells competent is they kind of poke teeny tiny holes, and I'm going to make these holes certainly much bigger than what they would be in reality. So what happens, those teeny tiny holes are induced into the bacterial cell. It's not enough for the bacterial cell to break open and lose its cell contents, but it is enough that that insulin gene now can enter our bacterial cell through one of those pores or one of those openings, okay? The bacterial cell will heal that back up, but in the meantime, what's happened, our bacteria has taken up that bit of circular DNA. It's taken up that plasmid. What happens then, this is where the recombination comes into play, okay? It is possible for the bacterial cell to exist just in this state, to have its genomic DNA and the little bit of plasmid DNA, and still, because you've got that bit of plasmid DNA, the molecules involved in generating RNA molecules and protein are still going to be able to read that plasmid and make that insulin gene, okay? But what can happen and what's more stable is if recombination happens. So if the bit of bacterial DNA kind of opens up a little bit and takes up the insulin gene, okay, This bacteria now is going to be our recombinant bacteria. And what's going to happen, that transcription and translation will happen, and it's going to pump out, essentially, lots of insulin. It's going to make lots of that insulin protein. But the bacterial cell doesn't need it. So what they do is they then harvest the bacteria and they extract the insulin protein from the bacteria. Okay? This is actually a much more stable source of insulin than what was used previously. Because previously, if you were diabetic, and my grandfather was diabetic back in the 1970s, the source of insulin way back in the day, they used to take pig pancreas, ground up pig pancreas, extract the insulin. Problem is, when you do that, the pancreas has all kinds of other enzymes associated with it. So there wasn't real consistent in as far as the dose of insulin that they were getting out of the pig pancreas. By taking advantage of bacteria like this and doing it in a recombinant method, now we're making lots and lots of that insulin protein, and we don't have to worry about it being broken down. So they're recovering much larger amounts of insulin, and that makes a product that's more stable for people who are diabetic. Okay, so I used insulin as the example. Insulin isn't the only protein that's made this way. Okay, human growth hormone is made this way also. So instead of inserting the insulin gene, they insert the gene for human growth hormone, and that bacteria cell now pumps out lots of that human growth hormone. Okay, so there are lots of different proteins that are made this way. Now, historically, I'm trying to say, the way this process was discovered was by a scientist, and that was Frederick Griffith. Okay, and Frederick Griffith did a set of experiments and what he was tracking in this case was a virulence factor. So what he had is he had bacteria that were smooth and bacteria that were rough. So the bacteria that were smooth essentially had a capsule that went around it. The bacteria that were rough lacked the capsule. Now, the key here in how this works, the smooth bacteria, when transferred to mice, would result in the mice dying because that capsule made the bacteria pathogenic. So making that pathogenic, that was the virulence factor. All right? So if he used that, he knew he got the mice to die. If he injected bacteria that were rough, that didn't have the capsule, the mice were perfectly fine, and he was able to recover that bacteria from the mice to show that yes, they did indeed have the bacterial infection, but it wasn't lethal. And it wasn't lethal because that particular bacteria wasn't virulent. It lacked that virulence factor. So what he did is he took the smooth bacteria, heat inactivated it, and injected that into mice. And the mice did fine because by heat inactivating the bacteria, he had killed it. So the bacteria weren't making the virulence factor. He also wasn't able to culture bacteria back out. So he was essentially injecting dead bacteria. Now I'm going to change colors here because the key in this process was taking the smooth heat, I'm going to abbreviate that just as HI, and you know that HI means heat inactivated bacteria, plus live rough bacteria. And what happened was the mice died, okay? So we knew from his experiments that just the rough bacteria wouldn't be lethal for the mice. The heat inactivated smooth bacteria wasn't enough to kill the mice. But you put them together, what happens? The gene for virulence, that gene for making that capsule, was transferred to the rough bacteria. And when he cultured out those bacteria that he recovered from those mice, instead of recovering rough bacteria, he recovered smooth bacteria, meaning that those rough bacteria now picked up that capsule, that virulence factor, and that was transferred to the rough bacteria. So that's what kind of historically gave rise to being able to transfer genes to bacteria intentionally, okay? Now, the experiment that Frederick Griffith did, the DNA wasn't on a plasmid. In this case, it was just a fragment of DNA that was picked up by the recipient bacteria, by that rough bacteria. It picked up just the gene for the virulence factor, but again, once it picked it up, that changed the phenotype associated with the bacteria. So it went from being rough to smooth. And as a result, he observed that the mice died. Okay? So it's really kind of a neat process how that was discovered. But what's really key about transformation is this really now has become a very powerful tool in the pharmaceutical industry, in the biotechnology industry, and at the basic research level as a way to make one particular protein. Okay? Hopefully that made sense. If you have any questions, please let me know.